Hello, I'm Jyotika. Welcome to 𝗖𝗓𝗟𝗟𝗢𝗓. In today's class, I'm discussing how you should prepare the method section for any of your AS level studies. I hope you're watching this video only after you've watched the previous one, which was about how you should prepare the introduction section for any study. So do watch that video because only then this video will make complete sense to you. I'm going to be using the Canli et.al. study as an example as I explain to you how you should be preparing for the method section. Also in the last video, I have addressed some questions that students typically have at the AS levels before they begin their preparation of the studies. So please watch that video. The only thing I would like to repeat from that video in this one is that all these videos that I'm making, this series of videos, is not meant for those who are looking for quick lessons or quick solutions. So if that is what you're looking for, please skip these videos because these videos are going to be lengthy. Okay, so now coming to the method section for any study, how you should go about preparing it. The problem I have noticed when students prepare for the method section is that they do not prepare equally the method section for all studies. And this is because, like I was telling you last time, in the companions, in one of the companions which is being recommended, there is arbitrary coverage of the study. So for some of the studies, the independent variable is explicitly outlined. For some of them, it's not even mentioned. So students also go about doing exactly that, what they find in the companion. That is not the approach that you should take. For each and every study, you have to prepare all the elements that are there in the study. So if for one study you're preparing the independent variable, you have to prepare it for all the other studies as well. Okay, so every study has to be prepared in the same way. Okay, and which elements you have to elicit from the method section of every study is clearly outlined in the syllabus. So there's no doubt about it. Again, refer to your syllabus. They have clearly stated which elements from the method section you need to prepare for. And those are exactly the elements that they ask about in the question paper. So once you do that, you can't go wrong. Okay, so using the Canli et.al. study as an example, I will tell you which sections you need to prepare for in the method section and how you can do that. So first, what I've presented here is the experimental paradigm. Most of the studies that you're having in your course are experiments. You know there are many research methods. One of them is an experiment. Most of the studies in your syllabus are experiments. So this is what we call an experimental paradigm. If it was not an experiment, the study, we would call it the general procedure. Basically, in the section of experimental paradigm or general procedure, what we prepare for is the general overview of how the study was conducted. So the procedure, just an overview of it, not the details of it. Okay, so after you've done one reading of the original paper with you, after that you just need to outline what were the steps, the broad steps in the study or the overview of the study, like I told you. So just list it out step by step. What you can ask yourself is what participants had to do in this study stepwise, and just quickly note those steps. Now, though this is not a part of the syllabus, and what they ask you in the question paper is details of the procedure which was followed. Nevertheless, when you are beginning preparing your notes, you need to list out this section of procedure or paradigm because it helps you to understand all the rest of the sections well. If you do not prepare for this section, if you first don't take an overview of how the study was conducted, the rest of the sections don't make sense to you because you don't know exactly what happened in the study from start to finish. So first, for any study, just note down, after you've read the study, just note down what were the broad steps that the study followed in its procedure. So here, for example, if we take the Canli et.al. study, the first thing we need to note for any study is whether it was conducted individually for every participant or in groups. Okay, for example, you have that 𝗬𝗎𝗍 et.al. study which was conducted in groups of 8. So for those type of studies, you can note that there was group testing. For this type of a study like Canli et.al., first only note down where you're preparing your notes that it was an individual testing of participants. This is important to understand what the participants went through in the study, and it also helps you later on to talk about ethics such as privacy. Okay, so the first thing for every study, just note down explicitly whether it was individually conducted for every participant or in groups. The next thing is what happened with the participants or what they had to do in the study, again stepwise from beginning to end. So if we take the Canli et.al. study, the first thing that participants were made to do and what they were made to undergo was that they had to undergo an fMRI scanner. Like we saw in the introduction section, we've already seen why the participants were undergoing an fMRI scan. So the first thing that happened with them in the study was that they were asked to undergo that fMRI scanner, and the whole procedure of the study was conducted inside that fMRI scanner. How it was done, we'll see very soon in details, but for now we just know this is the first thing that happened to participants. They underwent the fMRI scanner. In that scanner, there was some activity that they had to perform, and as they were performing that activity, their brain was being scanned, which is why they were in the fMRI scanner. If we talk about which activity they had to perform, again we are just speaking broadly at this point. They had to view certain photographs. Which type of photographs, how many photographs, we see very soon in the material section. For now, we just know that they had to view some photographs in the scanner, and they had to rate each photograph for its emotional intensity. Why this is happening, why they were made to do these things, you'll understand only if you have seen the introduction section. Okay, so this is what happened at the beginning of the study, and this can be clearly classified as session 1 of the study. Okay, because there's a time gap after this session has occurred. So we can clearly identify 2 sessions in this study. There are many studies in your syllabus that have taken place over sessions. So always note that, because in the paper also they ask you what happened in session 1 or what happened in session 2. So, okay, so clearly you should know exactly how much happened in session 1 and how much happened in session 2. Okay, so session 1 was about individual testing of participants in the fMRI scanner, in which their task was to view certain photographs and rate each of the photographs for its emotional intensity. This much you should know before you proceed with the rest of the method section. Then once you come to know exactly what the materials were or exactly which equipment was used with participants, you'll be able to fill in those details in this outline or the structure which you have created at this point. Next, obviously you have noted about session 1. What is the overview? Then you should note for session 2. So session 2, first you need to note when this session occurred. What is the time interval between both the sessions? So first thing we notice in the Canli et.al. study, this session occurred 3 weeks later. Okay, again there was individual testing in this session, and what was done was participants were asked to view a lot of photographs, some of which they had seen previously, meaning they had seen those photographs in session 1, they had rated those photographs. Some were new photographs, which they were seeing for the first time, in the sense that they had not viewed those photographs in session 1. Now what they had to do this time around was they had to rate those photographs they were seeing, both the old and new ones, as either having been seen before, that is having been seen in session 1, or they had to rate it as something new. So for each photograph which was presented in a random order, obviously you can understand that, they had to look at the photograph and just rate and indicate whether they had seen it in session 1 or they were seeing it for the first time. If they indicated that they had seen the photograph in session 1, they also had to indicate the amount of certainty they had that they had seen the photograph in session 1. So they had to indicate whether they were sure they had seen it in session 1, they were sure they had not seen it, or they were just familiar. They felt like they had seen it in session 1. So they had some amount of doubt. So this is what happened in session 2. Again, like I told you, I'll just remind you, this is the broad procedure, not the specific procedure. In the exam, when they ask you, you have to give the specifics as well. This, you are just preparing this section before proceeding further to have a framework in your mind with which you can understand the rest of the sections within the method section. Okay, so this is about the experimental paradigm, or the general idea of how the experiment took place. So always after one reading of the method section, prepare this section. Next, you can see you need to note the sample and sampling technique. This is what is prescribed in the syllabus, that when you're discussing the method section, first note down what was the sample and what was the technique of sampling. At this point, whatever you've learned in general in research method, like I was discussing with you last time, that is going to play a very important role, because this is the point from where we start evaluating every element of the method section, and that evaluation we cannot do unless we have understood that preliminary general section on research methods properly. In that section, you must have come across what are various sampling techniques, their general descriptions, which sample characteristics are desirable, which limit the generalizability of the studies. All that is explained to you in general without reference to any particular study. So only if you have learned that well and your concepts are clear, will you be able to use that knowledge of that preliminary section to evaluate the specifics of this study. Okay, so make sure that is very clear to you before you come here. I'll be giving you an example of how to apply that section here, but that will be only for the Canli et al. Study. For the rest of the studies which you're preparing by yourself, for those you should have that knowledge with you so that you can apply it to those other studies. Okay, coming to the sample and sampling technique, what you must have already learned in your research methods section is the sample refers to the participants of a study. Those people who are participating in a study and either something is happening with them or they are performing something or both of it, those are referred to as a sample of a study in psychology, because psychology is about the human mind and behavior. So its participants are going to be human beings for the most time. Coming to the Canli et al. Study, when we are noting the sample characteristics of any study, such as Canli et al., what all do we need to note? In your notes, first thing you should note down what was the total strength of the sample, which we denote with a capital N, like you can see here, simply meaning how many total participants were there. So in the Canli et al. Study, there were 10 participants, and on the screen you can see I have noted N is equal to 10. So whenever you're listing the sample characteristics, which you should, because there's a direct question in the paper, give the sample characteristics of this and this study, and you also asked to evaluate the sample characteristics many times. So first have them in front of you very clearly. Sample strength is one characteristic. Also, one more thing I would like to tell you right here before we discuss further. Many students, they are asked for 2 characteristics, they tend to write down all the characteristics of a sample. So be very clear of what constitutes a characteristic of a sample. Any one demographic of a sample is one characteristic of that sample. So if they're asking you for 2, you could give strength and gender, gender and age, strength and age, something like that. You don't need to give more than that. Okay, so if you're in the habit of listing down like this, you'll be very clear as to how many characteristics you're listing out. So this is very important in your preparation. So the first thing is you note down the total strength of the sample. In the Canli et al., there were a total of 10 participants. So I've noted it as N is equal to 10. Next, you can see you need to note down the gender. These are things you're going to find in all the studies. What was the strength? What was the gender distribution? In the Canli et al. Study, there's no gender distribution. There was a single gender that was tested, which is the female gender. In the research paper, as soon as they mentioned that only females were participants, they've justified why only females were participants. I'm not discussing that right now because I will discuss that in the control section. Okay, so for now, because it's about sample characteristics, I'm just focusing on that, and I have noted that all the samples, meaning all the participants, meaning all 10 participants were female. So first I've noted the strength, next I've noted the gender, next we note whatever additional information is provided in the paper. Sometimes it's going to be age, sometimes it's going to be some other characteristic. So for Canli et al., what has been noted is that all the participants were healthy. Okay, when it comes to brain scanning studies, particularly, you might note something like this, that they are healthy. Even in the 𝗧𝗪𝗧𝗧 study, you get to see that something is mentioned about the health of the participants. This is because the brain structure and brain functioning of individuals who are healthy as against those who are unhealthy is different. Okay, so it becomes important to note this. Okay, so the next characteristic, we see that all the 10 female participants of this study were all healthy. And the fourth characteristic that we see that all of them were right-handed. That is, their dominant hand for writing and other work was their right hand. So there are 4 characteristics mentioned in the Canli et al. Study. Always note down all the sample characteristics and cross-check whether you have missed out on anything. Next thing you need to do before proceeding with the sampling technique is evaluate the sample characteristics right at this point. Don't leave all the evaluation for later. Do not arbitrarily evaluate. So I'll evaluate only the variables of the study or only the research method of the study, and I won't do the rest. That will not do, because you asked specific questions in the paper. There can be a question asking you to evaluate the sample of the study. Then you'll end up skipping that because you did not prepare that section. Also, when you're asked for longer questions, that 10-mark question which comes on evaluation of strengths and weaknesses, there is always a named issue in that. That named issue, if it turns out to be generalizability and you have not prepared this section properly, you will not be able to comment on it. So you cannot pick and choose as to which elements you want to evaluate or which parameters you want to evaluate them on. So on some studies you just want to consider generalizability, on other studies you want to consider validity. This is not how you can go about. You have to prepare each and every type of evaluation for each and every type of element in every study if you want to attempt 100% of the paper which comes. So coming to evaluation of the sample characteristics, whenever we evaluate sample and sampling technique, the main question that we address is how generalizable are the findings of the study. So sample characteristics and sample technique have direct bearing on how generalizable findings are. One thing that has bearing is settings of the study, but that is something we consider in research methods. When we evaluate the research method, which I'll do in the next video. Today, in this class, I'm not going to evaluate the research method because that is a big topic in itself. There, we consider the settings also, how generalizable the settings are. Here, we just consider how generalizable the sample is. So looking at all the characteristics now, how we need to evaluate is, we've already learned about what generalizability is in the research method section before beginning with the studies. Now we pick up one sample characteristic at a time and evaluate according to what we have learned how generalizable that characteristic of the sample makes the findings of the study. So for example, when we see n is equal to 10, we know this is justified. A study like this will have a small number of participants because brain scanning is very effortful and it takes a lot of, it's very costly also. So in such studies, typically we do have fewer participants, but from what we have learned in our research methods section in the beginning, we know that small samples limit generalizability. Okay, we cannot be very confident in how much we can generalize when we have small samples. So, or immediately when we see this is one characteristic in this study, that the sample size was small or the strength was small, immediately we know that this generalizability, overall generalizability of the study has been limited. Okay, so like this, characteristic by characteristic you can evaluate and you can note down whatever you're evaluating in two columns. One should be strengths, one should be weaknesses. Obviously, if you're saying generalizability is limited, that will go in the weakness column. Okay. The next characteristic, we saw all the sample, the entire sample in terms of gender, it was all females. Immediately again from what we've learned, we understand that this limits the generalizability to males. We are not saying, understand this, we are not saying that necessarily we cannot generalize to males from this sample. It is very much possible that what is true for females is also true for males. Okay, need not be different. But all we are saying is, since that is something that we have not investigated in this study, we cannot be certain that what has been found with females in this study will be applicable to males. We will have to do a separate set of study with males to be sure about that. Okay, so what we say is generalizability is questionable or generalizability is limited. Do not jump to the conclusion that it is not generalizable to males. It could be, we don't know. Okay, so indicate when you're writing your answers, write in a way that shows your understanding. So indicate in your answer that the generalizability is doubtful or questionable to males. So this is a limitation of the study, limited generalizability. Does not mean it's a fault of the study or it's a problem with the study. It's a limitation. It's something that restricts the extent to which the study is generalizable. Next, we see all the participants were healthy. This is a positive in the sense that we know that the large section of the population, majority of the population is healthy. So it's going to be generalizable to a large number of people. It is limitation also because then it's not going to be generalizable to those people who are having specific disorders. We are not sure whether these findings are generalizable to those people or not. Okay, so it's both. We always take a balanced view. If there's some strength, we note that also. If there's some limitation, we note that also. Okay, also all participants being healthy is a plus because they're likely to perform the experiment with full sincerity. They're in the frame of mind that they can perform the experiment without any hassle. So that is also a strength of a healthy sample. Then we see that the entire sample was right-handed. Again, this limits generalizability to right-handed people. From whatever we find in this study, we'll only be sure to a certain extent that whatever results are there will be applicable to females who are right-handed. We cannot say whether it will be applicable to females who are left-handed or males who are left-handed. So again, that is a limitation. Okay, so that is about the sample characteristics. I hope you've understood how to prepare. List out all the sample characteristics. Consider each characteristic in terms of how much it enables or restricts the generalizability of the study. Next, we see the sampling technique. Here, I think it's clearly mentioned, I don't remember, but I think it's clearly mentioned in the paper that it was a volunteer sample. Okay, sometimes it's not directly mentioned which type of a sample it is, but you can infer from the details which are given. Like in some studies, you might see that the researchers belong to a particular university. I think this is in the Bandura study. They belong to a particular university and the students have been recruited from some nursery school affiliated to that university. In that case, it makes it very obvious that it's an opportunity sample. Again, how will you be able to decide which type of a sample it is? If you've done your initial research methods section, then you'll be able to decide. So here, I think it was specifically mentioned that it was a volunteer sample. So we don't need to think much about it. A volunteer sample, you would remember from your research method section, is a self-selected sample. Means simply it is people who agree to be a part of the study, who are keen to be and who are interested in being a part of the study. They are forthcoming. They come by themselves to the experimenter upon some advertisement or some declaration by the experimenter, some announcement. They come by themselves and tell the experimenter they would like to participate, and then they participate in the study. So that's a voluntary sample. Now, to evaluate this sampling technique, again, how will you be able to do that? If you have prepared your initial section well, because in that, in general, without reference to any study, they have told you what are the pluses and what are the minuses of a voluntary sample. So if you've understood that well, you will be able to apply those positives and negatives to the sampling technique of this study. Of course, you have to make it relevant. Here, I would like to bring about that very big problem which is there with most students' answers in the paper. They give very, very generic answers. Okay, so they have learned what the positives and negatives of a voluntary sample are. They have learned that in the Canli et.al. there's a voluntary sample, and then they will just list out those general strengths and limitations of a voluntary sample instead of making it relevant to the study. That is a very big mistake, and that is why if there are 2 marks, say for an answer, you just score 1. Because 1 mark is always reserved to check your understanding, whether what you've learned you're able to apply it to the study or not. That's the ultimate goal. The goal is not to make you memorize a lot of strengths and weaknesses or a lot of descriptions. Okay, so coming to the sampling technique, it was a volunteer sample. In general, we have learned that a volunteer sample is easy to recruit. So here also I've listed out the general points, but I'll explain to you how it is relevant to the study. We say that a volunteer sample is easy to recruit. Now you need to consider which aspect of the study made it necessary that we should have easy to recruit participants. This is how you should think. So which aspect? Do you think many participants, many people would agree to be a part of studies in which their brain is being scanned? No, people are fearful of such technology. Do you think many people would want to be in that tunnel and carry out the tasks? A lot of people are even claustrophobic. They would not want to be in that tunnel for long. And like I told you, a lot of people are scared of undergoing such type of technology. They feel that they might be harmed in the process. So for such a study, it is difficult to recruit participants. This is how you make it relevant. So you say that a volunteer sample is easy to recruit, and then you connect it to the study and say, because this was a study of the biological approach involving brain scanning, it would be difficult to recruit participants who would agree to carry out the tasks in the fMRI scanner. So a voluntary sample was very suitable to this study. This is how you make the strength relevant to the study. Next, you can see in general, we know voluntary samples easily engage in challenging tasks. Okay, you give them something difficult to do and they are ready to do it because they have come out to participate in the study out of their own eagerness to participate. Okay, so they are easily engaged in difficult tasks. You know, this is the general strength. Now, what are you going to ask yourself? What was a challenging task in this study which became easy for the researchers to conduct because there was a voluntary sample? Okay, so this should be your line of thinking. So here, when we are saying that it is easy to engage them in challenging tasks, in this study, the challenging task was again being in the tunnel, lying there for a period of time, watching images and rating those images. And those images we have already seen, they were emotionally intense, emotionally disturbing at times, okay, to emotion arousing, which makes people uncomfortable, which puts them at a state of discomfort. Those type of images were there. So it was a challenging task at a psychological level. Physically, you might feel there's no big deal in it, but psychologically, certainly you would agree that it was a challenging task. So for this type of a study, again, a voluntary sample was very helpful. This is how you should note the strength in your study. For every element, because I'm explaining to you how you should prepare, I have listed out number of strengths and weaknesses. You might not find it necessary to list out so many. So even two strengths and weaknesses, or one might suffice. Again, I told you these videos are going to be lengthy and I'm going to consider many things. So I'm just explaining to you, not necessary you note down every strength and limitation for every element, and not necessarily every strength and limitation in general of any element will be applicable to every study. That is not necessary. The next strength of a voluntary sample is that it is free from the bias that we see when researchers select a sample. When a researcher selects a sample, which is the case in an opportunity or convenience sample, at that time he tends to knowingly or unknowingly, mostly unknowingly, because researchers have good intentions, they don't want to spoil their own study. So unknowingly they tend to select participants who would act in ways that would be favorable to their hypothesis, who would help them prove their hypothesis as being correct. This is a problem with opportunity samples, which gets eliminated in voluntary samples, because here the researcher does not select the participants. It is the participants who themselves come forward to be a part of the study. And for the Canli et al. Study, now what you need to consider, what was the hypothesis? It was the hypothesis was that emotional memories would be recalled better than non-emotional ones. So now coming to how researchers could have had a biased selection in the sample. If they had a chance to select their own sample, if they had decided to select their own sample, they might have selected people who are more emotional, because they would expect the amygdala to be more activated, so they would get clearer results and stronger results for their study. That was a possibility. Or simply sometimes researchers select those participants who are more willing, more vulnerable, more suggestible. So, who are easily given to the demand characteristics of the study, they tend to select those type of participants. If we talk specifically about Canli et al. Study, they might have selected participants who were more emotional by nature if they had a choice to select. That is, if they had taken the choice to select. But they did not do that. So we can be quite certain that their bias was in no ways involved in the selection of participants. Next, we look at the limitations of volunteer sampling. In general, we know volunteer samples share certain characteristics. What I mean by influential characteristics is that which could influence the results of the study. So obviously people do share a lot of characteristics, but all characteristics do not influence the results of a study. Certain characteristics can. Now, if we look at a volunteer sample, maybe they are more emotional by nature. That's a possibility. Okay, because they are more emotional, that's why they volunteer for studies, because they think it's nice, it's good, it's nice to help people. They have these type of moral beliefs, and they have kindness in them, which is a very positive emotion. So they have this kindness and generosity, which is why they participate in the studies. Now, this study, Canli et al. Was about emotions. If the possibility is there, which is there, that volunteers are more emotional by nature, that could have a bearing on the results of the study, because the way they are going to process those images and the intensity they are going to find on these images is not going to match with other people who are less emotional or who are more rational than them. So these type of problems can come in a voluntary sample. Again, in general, you know that they share certain characteristics, but when you're writing for a particular study, you need to mention which characteristic, you need to give some example, which characteristic they might have shared, which would have had a bearing on this particular study. So here, like I told you, for the Canli et al. Study, if they shared their emotionality, that could have been a problem for this particular study. Besides that, we know volunteer samples are self-selected. Self-selection is a problem in itself, because that is just unrepresentative. When you've learned about sampling techniques, you have learned that random samples are representative. Frankly speaking, no other sample is representative. So other samples are not desirable. They're just there for practical convenience of researchers. Okay, so random samples, they represent the entire population, voluntary samples do not. Now, when it comes to this particular study, when it comes to how emotional people find stimuli or how they remember emotional memories in the whole population, if you take in the whole population of a country or the world, people have a lot of different ways of emotional processing. You know, people are not emotional in the same way. People have their unique emotions. Some might process some emotions more deeply. So some people, like we say, they are negative, so they process negative emotions more deeply. Some process positive emotions more deeply. There are a lot of variations in people when it comes to emotional processing, when it comes to emotional activity, emotional memories. Those we can accurately represent. All these variations which are there in people, we can accurately represent only when we have a random sample. Not when we have convenience type of samples, meaning opportunity or voluntary samples, because those, like we saw earlier, they could share certain characteristics because they're self-selected, and that can become a problem. Here, when we are saying they're self-selected, the problem is they're not representing all the variations which are there in emotionality in people. Okay, so that is a problem for representativeness. Whenever there's a problem for representativeness in a study, automatically generalizability becomes restricted. Okay, so that is what we are noting here. So this was about the sample and sampling technique. One more thing I would like to tell you from what I have learned with my experience with students before we proceed is, students tend to mix up sample characteristics and sampling technique. Please consider them separately. They are not the same thing. Okay, so sample characteristics have their own pluses and minuses. Sample techniques have their own pluses and minuses. Please evaluate them separately. Next, we look at the materials of any study. Materials are directly intertwined with standardization. Actually, standardization is seen at two levels, material and procedure. But first, so that's why before going to standardization, we should consider materials. Procedure overview we have already seen. Also, once now we see the material of the study, we'll get quite a good idea of the specifics of how the study was conducted. So let's consider the materials in a study. In general, when we are talking about materials, you need to look at each and every piece of equipment, each and every tool, each and every object that was used in a study. Okay, so whenever, like if you're reading your original paper, whenever you're coming across any object that was used, any tool, any equipment for measurement, for manipulation, for whatever purpose, just note it down under a section called materials. Also, how you note it down is not just the name. In its full detail, you need to note down. Okay, and yes, again, there are direct questions because a lot of students are just concerned with the exam. So yes, there are a lot of direct questions in the exam from material section. They could ask you for some examples of materials used in a study. When you're answering that question, you need to note the material in its entirety, in the sense all the details that have been given about the material you need to note. So that I'll tell you how with the help of the Canli et.al. study. Here, the first material, which is very obvious right from the beginning, an fMRI scanner was used. Now, can I just say fMRI scanner was used? No. Whatever details have given to me about which type of fMRI scanner, what were its properties, those also I need to note down. Again, in the paper, you might find a lot of technical details. Okay, so if you're getting confused exactly how many of these details you need to know, in that case, you can refer to your course companion and check how much is covered in that. Otherwise, it is pretty obvious. When you're reading the paper, as a psychology student, how much you need to know, you don't need to know like a technician. You don't need to know about every component of that fMRI scanner. So those details you can ignore. Just the overall main properties of the fMRI scanner which I mentioned, those you need to be able to put down in your paper. So here you can see there was an fMRI scanner. MRI scanner, fMRI scanner is the same thing. The machine is the same. So you can call it either ways, with a 1.5 Tesla magnet. 1.5 Tesla magnet, you don't need to explain, but just to explain to you, it has 30,000 times a more powerful magnetic field than the gravity of the Earth. So very, very powerful. What we are indicating by 1.5 Tesla magnet is that a very powerful magnet was there in that machine. Obviously, the more powerful the magnet, the better it is able to detect the magnetic properties that are there in such fine elements inside the body. So a very high quality, very sophisticated fMRI scanner was used. In that fMRI scanner, we also need to note what type of a mechanism was placed for participants to be able to do their task. because that has direct bearing on what we are learning in this study. So there was a back projection screen and a button response input mechanism. A back projection screen, participants had to view images in the fMRI scan. Now, how would they view those images? You can't give them an album in their hand and ask them to turn pages and view images. Okay, they are in a very still, static state inside the fMRI scanner. And we don't want too much movement because one, they could be harmed by too much movement. Second, they could be the results could be confounded. If they're moving a lot, it's difficult for the scanner to detect properly, to scan their brains properly. Okay, so obviously you need some place, some screen where they could look directly at the images rather than giving them something in their hand. So there was a back projection screen for that purpose inside the scanner, so that when they were lying down inside the scanner, they could look at the images which were being flashed on the screen. And we know that they had to rate each image. We had seen that in the procedure. Now, how would they rate it again? We cannot expect them to write down or speak inside the scanner. So then there was a button response input. Okay, they had to press some button. There were like 4 buttons, each button indicating one level of intensity. So there was a button suggesting 0 emotional intensity. If the participants felt that the images were not emotional at all, they had to press the button for 0. If they felt it was slightly emotional, for 1. If they felt it was even more emotional, for 2. And if they felt it was very emotional an image, they had to press 4. So for every single image, they had to give a rating by pressing a corresponding button. So obviously the screen mechanism and the button mechanism both were there inside the fMRI scanner. As we are seeing the materials, not only do we need to understand and learn about them, we also need to connect them with that framework of the procedure we have created in our mind. So in the paper, if they're asking us about the procedure, what did participants have to do in session 1? I told you can't just give an overview. So these are the details you need to add at that step. So you say in session 1, they had to go and undergo an fMRI scanner in which there was a projection screen and a button response mechanism. This is how you keep on filling in the details there. Okay, why I have not taken it there and put it separately under material section is because you should know clearly the distinction between the procedure and the materials. When they ask you in the paper, you should be able to list out materials and not confuse it or intertwine it in the procedure. Okay, so the fMRI scanner was the first material used in this study. Next is the pictures which the participants had to view. They were not random pictures. They were not just any pictures being flashed on the screen. They were pictures taken from Lang and Greenwald's 1993 set of an International Affective Picture System Stimuli set. These were highly standardized images. They had been used in previous study. Each image was known to have a certain emotional value. How it was known to have a certain emotional value? Because these images had been used with several participants across studies. These several participants had rated these images. And so how people typically perceive these images had been measured in past studies and it had been noted. So this was a standardized set of pictures called the International Affective Picture System Stimuli set. So this name is easy to remember. It's long, but affective meaning emotional, so it becomes easy to remember, and it's been used internationally. 96 images from this set were taken for this present study, session 1. So for session 1, 96 images were drawn from this standardized set and it was flashed to participants on the screen as they lay on the fMRI scan. Okay. Again, why you need to note these things is because we don't evaluate materials as such, but when we talk about standardization, how standardized the procedure was, indirectly we are evaluating the materials, because materials which are sound, well established, we include them under standardization, which are not, we don't include them. And standardization by itself is a very big plus for any study because it makes the study highly reliable. Again, this is something you've learned in your research methods section. Okay, now we need to know something more about the standardized set of images which was used for this study, from the pool from which the images were drawn. One thing is that in this set, the International Affective Picture System Stimuli set, in this, all the photographs had some ratings associated with them. So each photograph had a rating associated with it. These ratings were in terms of 2 dimensions. Okay, which are those 2 dimensions? When it comes to emotions, emotions can be understood in terms of 2 properties. One is arousal and one is valence. When it comes to arousal, I think it's very easy to understand because arousal just means, last time I told you emotions are psychophysiological responses. So arousal just means how much it arouses your body. Okay, so one property any emotion has is how much it arouses your body, how much it puts your body into movement, or how much it mobilizes your body. So that is one dimension of an emotion. Another dimension is its valence. Valence can be understood in many ways because it simply means value. But in this paper, if you read it, it's clearly specified valence refers to the degree of negativity of an image. Okay, so here higher the valence of an image, the more negative it was, and lower the valence, the more positive it was. Okay, you know emotions are negative and positive. So here we are talking about that property of an emotion. So in this standardized set, which was from which photos were drawn from this study, each image had a rating associated with it in terms of how emotionally arousing it was and how much emotional valence it has. This is very important for the study, we'll see ahead. But we need to note this at this point. Now, there have been questions in the past asking the range for the valence and arousal, or the correlations for valence and arousal, which we'll see ahead. So these figures are very important. Here you can see these normative ratings for valence ranged from 1.17 to 5.44. Okay, so here I'm sorry, the mistake I made is the degree of negativity. So the lower the score on valence, the more negative the image was, and the higher the score, the more neutral it was. That is the interpretation. So I'd just like to correct that. Okay, here you can see, firstly, what do we mean by normative ratings? Like I told you, these images have been shown across hundreds of studies to participants, and in each study, participants have rated each of these images. So that is what we refer to as normative ratings. Normative are just comparative ratings. So for every image, we know how participants of different studies have rated them. Based on that, we have an average of how that image is typically rated. In terms of valence or degree of negativity in this set from which the photos were drawn, those 96 photos are being spoken about here, which were drawn for this study. So in these 96 photos used in the Canli et al. Study, the valence range was from 1.17 to 5.44. That is, if we consider all the 96 photos, the photo having the least valence was having a valence normative rating of 1.17. The photo which was having the most valence in these 96 photos was having a rating of 5.44. Okay, so you need to know about these figures also. Again, talking about these 96 photos itself in terms of arousal, because we saw that is the other dimension. This arousal ranged from 1.97. So the least arousing photo in this set of 96 had a rating of 1.97, indicating it was a tranquil image. What is tranquil? Very calm or peaceful. So when participants viewed these images, they did not feel aroused at all. So those were the images on the lower range of the arousal ratings. And then there were images on the higher end, with the highest end or the maximum range being 7.63. So the photo that was the most arousing in the set of 96 had been rated normatively in previous studies as being 7.63. What we come to know from this for our understanding is that there was a wide range of photographs used in this study in terms of both dimensions. So in terms of valence also, a wide range of pictures were used. In terms of arousal also, a wide range of pictures was used. This is a very good element for any study, because if such a good range is used, we get really good results. Okay, because our results are not restricted in any way to a certain range. They are across a wide range. So we come to know a lot about how the amygdala works and how people form memories at different levels. Okay, which was a very big concern of the aims of this study. Okay, then coming to 48 foil images. What are these foil images? When we saw the general procedure, we saw that the study was conducted in 2 sessions. In session 2, there were some extra images which were not seen in session 1. If you think logically, you'll be able to understand why a researcher would include extra images, because he wants to check whether participants really remember what they've seen in session 1 or they're just guessing. So he can compare the images, their ratings for the images they've really seen, their memory ratings for those, and their memory ratings for new images, and see whether they're just guessing as having seen photos before, or they really can distinguish between photos they've seen before and they haven't. Okay, so that's why there were some pictures, new pictures included in session 2. These have been referred to as foil pictures. What is a foil picture? Meant to confuse or meant to mix up with the previous photos and distract the participant, those type of photos. Now these photos were also drawn from the same International Affective Picture System, and you can see even their valence and arousal ratings have been presented. So now for these 48 foil images which were used in session 2 as new images not seen in session 1, for these 48 the valence normative ratings range from 1.31 to 5.78. so the least photo with least valence was having a rating of 1.31. The one with most valence was having 5.78. Others were in between. And the arousal ratings from 2.74. The least arousing photo was 2.74. The highest one was 7.22, highly arousing. Okay, now these figures do look different from the ones we've seen previously, but not markedly different. How researchers decide whether two sets of pictures are comparable or not is they do some statistical significance testing. Again, significance testing, something you've learned about in research methods section. So significance testing they do and they compare. So in this study also they had done that. They had compared the original photographs and the foil photographs for their comparability. To what extent they are similar on these dimensions of valence and arousal. Obviously that is important because otherwise it becomes a confounding variable. If my original photographs are having different properties and my foil photographs are having different properties, then participants will give different ratings to these photographs, not on the basis of whether they remember it or not, but because of the unusual properties that the new photographs are having. So that's a confounding variable. So we need to make sure that foil photographs and actual photographs are comparable, and for that the researchers do statistical significance testing. So even if the figures look a little different here, upon comparison they're not exactly the same. Despite that, you can be sure that the researchers had very carefully made sure that the foil photographs are comparable to the original ones. Again, something we'll come back to in the controls. As of now, we just need to know these were the materials used, and this is how you need to list out materials with their complete details. Next, we have standardization of the study. Coming to standardization. Standardization in any study refers to what extent the procedure has been kept the same for every participant of the study. It is very important. For a study to be reliable, standardization is very important, because only if the procedure has been kept similar across participants can we say that it is not the way the procedure has been conducted or the way the material has been presented that has influenced results. Rather, it is the participants' performance or measurement which is influencing results. So again, you've learned this in the preliminaries. Coming to standardization, like I told you, both material and procedure account for standardization. And if we look at the larger meaning of standardization, it also means not only in one particular study how much a procedure or material have been kept constant, but like we just saw one example of the International Affective Picture Stimulus Set. Across studies also standardized materials can be used. These questionnaires which you get, like many of you doing your IPQ, find questionnaires say on emotional intelligence or which are those popular variables which you all study, job satisfaction, because a lot of you all are interested in the organizational sector, so job satisfaction, leadership. So many of you use those questionnaires which you'll find readily available. How come they're readily available? Because they're standardized questionnaires which have been used across studies and have been proven to successfully measure certain traits which you all are measuring. So that is also a larger way of looking at standardization. Coming to standardization for a particular study. So when you're making your notes, how do you note down which procedures or which material have been standardized? Again, after you've read the study, you know which of the procedures were held constant across participants or which materials were similarly distributed across participants. So keep noting that, and that you'll be able to do even better, like I told you, once you've written the procedure section and the material section. So let's consider which elements were standardized in this study. Also, one thing, there's a lot of confusion among students many times between standardization and controls. So when I come to the control section, I'll explain to you what's the difference between standardization and controls. Coming to standardization, the first way we see the Canli et.al. Study was standardized was that the order of photographs was randomized for every participant. Now, if it was randomized for every participant, why am I saying that it was constant? Because the order was not exactly the same for all participants, but the method of procedure is constant. Every participant is getting a randomized set of pictures. It's not like one participant is getting a randomized set of pictures, another is getting it in order. Okay, so the procedure of randomization was constant across participants. What you need to do is do some counterfactual thinking to understand why standardization is important. What if for some participants pictures were randomized and for others they were non-randomized, presented in certain order? Then what would have happened? That would have confounded results. Okay, because some participants would have been influenced by the order of the pictures rather than their emotional value or arousal. Okay, so that is a very important aspect, that for every participant, the photos were randomized. Next, you can see each picture was presented for a period of 2.88 seconds to every participant. When a researcher notes that he has presented certain material for certain duration, it is very precisely presented for that duration. So it's not roughly 2.88 seconds, it's precisely exactly 2.88 seconds. And the researcher can assure that it is for 2.88 seconds because it was on the projection screen. So you can time for how long every photograph has to be shown in that machine, in that mechanism, right? So the researchers showed every photograph to every participant for exactly 2.88 seconds. Again, why? Because otherwise if participants were shown the photographs for different durations, someone's shown for 2 seconds, someone's shown for 3 seconds, another is shown for 5 seconds, obviously that would influence their results, right? So that's why it had to be standardized. So reason for standardization is same across all. We just need to understand what all has been standardized. Next, you can see there was an interstimulus interval of 12.96 seconds during which every participant viewed a fixation cross. Why they viewed a fixation cross is a matter of control. We'll see it later. But the timing is very precise. So again, it becomes a part of standardization. Also, every participant saw the same thing in this interstimulus interval, which was a fixation cross. Next, you can see each participant was instructed to view each picture for the entire time that it was displayed. So this instruction is a very standardized instruction given to every single participant, that you must view the picture for the entire time it's being shown, because that is important for brain activity. Next is, after a picture's replacement with the fixation cross, the participant had to press a button. This procedure was also same for all the participants. After they had seen the photograph for the entire time, only then they had to press the button and indicate how emotionally intense they had found the picture to be. Next, you can see each participant had to choose from 4 buttons on the scale which I explained to you earlier also, 0 to 3. So every time the participant saw an image, he had to indicate from one of the 4 buttons, 0 indicating not intense at all, he didn't find the photograph at all intense emotionally, 3 indicating that he found it extremely intense. Note one thing, participants were not told to rate photographs in terms of arousal or valence, okay, because that would be too confusing. So they were simply told that you have to rate it in terms of emotional intensity, that valence and arousal characteristics, how they play a role, we'll see later also. But that was the basis of selection of the photographs. The basis of rating of the photographs was just that the participant had to see how emotionally intense he found the photographs and rate them accordingly. Okay, again, one more thing here, for paper preparation, you don't need to note so many points for standardization. Okay, you don't need to note so many elements which were standardized. 4 at the most, I think, suffices for preparation for the paper. Here again, because I'm explaining to you, I have noted several elements which were standardized. Okay, next you can see during the recognition test, that means in session 2, that memory test, recognition is a type of memory. So in the recognition test, participants had to indicate whether they had seen each photograph before. Okay, for the photographs, we noted this in the procedure, which they had seen before. For those photographs, they had to indicate whether they could remember with certainty or with familiarity that they had seen the photograph before. So first, for every participant, it was very standardized. First, they had to look at a photograph in session 2 and indicate whether it was new or old. If they said that it was old, they then had to say whether they remembered, that is, they were certain that it was old, or they just felt. So they were just familiar with the photograph. Okay, so these all are standardized procedures in any experiment. Experiments are very standardized, especially laboratory experiments. This is a laboratory experiment. Why I'm not discussing that aspect right now is because I told you research method video I will put up separately. This is a laboratory experiment, so lots of examples are there of standardization. Even I might have missed on a few over here, though I've tried to be exhaustive in this section. So you will find a lot of examples. As long as they are correct, you can note them down. Okay, next we come to variables. Very important. Variables are very important for two reasons. One is the entire study is based on manipulation and measurement of variables. The other thing is that for variables, variables determine, the independent variable determines the experimental design, which is another important element, and the variables determine which controls need to be kept. So a lot of things are associated, a lot of elements are associated with the variables. A variable, just to revise, is anything that takes on multiple values. In any study, we have two types, or we can say three types of variables. One is an independent variable. There can be many independent variables in a study, but one type of a variable you find in a study is an independent variable. In the case of an experiment, an independent variable is assumed to be the cause of an effect. So the causative variable, presumed causative variable in an experiment is called an independent variable. Effect variable is called a dependent variable, and then there are what we call control variables, sometimes just referred to as controls, but they are also variables. Okay, I won't go into a lengthy discussion of what variables are. You should be knowing that. We need to identify the independent variable of any study first when we talk about variables. Sometimes some students find it difficult to identify the independent variable. Okay, here I would like to note one thing. I know most of you will pick up from the companions or search online and find the independent variable for every study, but please make it please develop the skill of finding the independent variable by yourself. If you find it difficult to note what is the independent variable of a study, first you can note what is the dependent variable, then it becomes easy to note the independent variable. So I'm assuming you're just reading the original paper, you don't know which is the independent variable, which is the dependent one. You're trying to identify by yourself. So first, identify the dependent variable. Now, how do you do that? Very simple. You ask, what did the researcher measure in this study? That is the dependent variable. Once you've answered this question, what did the researcher assume was influencing this dependent variable? If it's an experiment, what you ask yourself is, what did the researcher think was causing this variable? Then you get your answer for what is the independent variable. So this is how you can go about identifying variables. Another thing which is very important and which students just neglect is the operational definition of variables. So I'll come to that also. But first, let's begin with identifying an independent variable. If you're trying to identify an independent variable directly, we just say for an experiment, we just ask ourselves, what is the presumed cause of the effect that the researcher is interested in? We know the effect the researcher is interested in from the aim. So for Canli et al, from the aim, we know researcher was interested in emotional arousal and emotional memories. So what did the researcher presume was the cause for this effect? So obviously he presumed that the emotional intensity of images is responsible for how aroused the participants feel and whether they develop memories or not, right? So that becomes the independent variable. It's as easy to identify the independent variable. Independent variables always have levels, as you have learned. So because they are manipulated by the researcher decides how to present the independent variable to every participant, and he can present every independent variable differently to the same participant, as in this study. Okay, so every participant was shown emotionally intense pictures, which was the independent variable, but at different levels. Sometimes the pictures were very emotional, sometimes they were less emotional, right? So those become the levels of the independent variable. Always in the paper, when they ask you to identify an independent variable, to outline it or to identify it, you have to state the levels. That's not an option. You don't just state the IV and leave it at that. You always note the levels of the IV, otherwise your answer is incomplete. An IV has to have at least 2 levels. That's for sure. At least 2 levels will be there. In this study, there were 4 levels. So you need to mention those. So in this study, the IV was emotional intensity of the pictures manipulated at 4 levels, 0, 1, 2, 3. As such, you would feel by looking at the procedure that it was not manipulated, but it was measured, which has been a criticism of this study very often, that it is correlational in nature rather than an experiment, because the researchers did not manipulate how emotionally intense the participants found the picture. The participants themselves rated whether they found the pictures very emotionally intense or less emotionally intense. But what you need to realize is it was definitely manipulated because the researchers had selected the images. We saw that was very important. They had selected the images which were known to have certain valence and arousal characteristics, and based on that selection only, participants also gave similar ratings. They also perceived the images in the way that they were selected. Okay, so if researchers had found a few images which were high on valence and low on arousal, participants did give a rating of 0 or 1 to those images. So basically, participants' ratings correlated with or corresponded with the way the images were selected. The researchers selected the images and presented them to participants. So definitely they manipulated how the participants perceived the pictures. So though it appears like participants rated the images, and so it was a measured variable, it was not, because how they rated the images was determined by the selection that the researchers had done. So it can be considered, you should consider it as a manipulated variable. Okay, so participants' ratings of intensity, though given by themselves, how much they perceived the intensity in a picture was determined by the manipulation of the researcher in the form of selection of the pictures. So it is a manipulated variable. In conclusion, we will say it was a manipulated variable. Okay, so it was manipulated at these 4 levels. We saw the meaning of these 4 levels. We've seen 0, 1, 2, 3. Now, coming to operational definition, very important and which students somehow find difficult, which is not. Operational definition of any variable just means how this particular variable has been put into operation in this study, how this particular variable has been employed in this study, how this variable has specifically been used in this study. That is what you need to ask yourself. See, if we have something like emotional intensity of pictures, as a researcher, I'm manipulating emotional intensity of pictures. There are so many ways in which I could manipulate it. Okay, so I could manipulate it. If I had to follow a rating scale only, I could manipulate it from 0 to 10. I could ask participants to give ratings from 0 to 10, why only 0 to 3? That could be my choice. I could have some justification as a researcher as to why I feel the scale should be from 0 to 10. I could not use a rating scale at all, and I could just ask participants to verbally tell me how emotionally intense they found the images. I could not use any type of self-report measurement at all, and I could measure participants' arousal, not through an fMRI, maybe through a biofeedback mechanism. I could just look at their gestures or facial expressions to determine how emotionally arousing they were finding the images. So to determine any variable, once we've identified what the variable is, like here we've identified emotional intensity of pictures as the variable, to manipulate it, there are many options that a researcher has. He can manipulate a variable in many ways. But he chooses one particular way in his study. Like I told you, he could choose a larger rating scale. He could have chosen a 2-point rating scale. He could have chosen a behavioral measure rather than a self-report measure. He has so many options, but he chooses one. For that same variable, different researchers are going to employ different ways of manipulating it. What you need to ask yourself is, when you're noting the operational definition, is, in this study, this particular researcher, how did he use the variable which was identified? And then very specifically to the study, how the variable was employed, you need to note that in the operational definition. So here you can see the operational definition is presentation of pictures of varying levels of emotional arousal and violence to participants, like we saw, resulting in perception of 4 levels of emotional intensity ranging from. Okay, so how these researchers operationalized the variable? They presented photographs of different arousal and valence to participants, like we have seen, and that determined the participants' perception at 4 levels, from 0 to 3. So this was their operationalization. Their rating scale of 4, that was their choice, and the photographs selection from that international affective picture system, that was their choice. Someone else could have selected photos from somewhere else. Someone else could have selected prepared his own photos for the research, which had never been used earlier. Okay, so whatever specific choice the researchers have made in this case, we note that in the operational definition. Again, an IV is associated with levels, so the operational definition of the IV also has to speak about its levels. And because we are speaking operationally, even the levels we have to state more specifically. So when I have just noted the IV, I have just noted that these were the levels. But when I'm saying operationally, I need to define what every level meant. So 0 meant not emotionally intense at all. 3 meant extremely emotionally intense. And the numbers in between indicated some intensity in between. Okay. So this was how intensity was operationalized. Again, if you look at the levels, there are different ways of manipulating the levels also. 0 could have meant very emotionally intense. I can do that as a researcher. I can take 0 to mean very emotionally intense and 3 to mean not intense at all. That's my choice. Okay, so again, this is operational in nature. Researchers decide how to use the levels of the IV, so we note that also in the operational definition. Then coming to the strengths and limitations. For every element, we have to look at the strengths and limitations. We have noted that. How do we look at the strengths and limitations of an IV? An IV is manipulated by a researcher. So there can be many things that account for its strengths and limitations. Okay, ethical considerations can be taken. Ecological validity will be a consideration. Any demand characteristics, social desirability, a lot of things apply when we take the strengths and limitations of an IV. Okay, so IV, but the main thing is validity when it comes to IV. See, IV, DV, they are part of psychometrics, something you see in the next year A levels, an issue of psychometrics, but you should know about it from now because it indirectly they do ask you about it in your AS level papers also. Psychometrics simply refers to measurement in psychology. So your IV and DV, more importantly the DV, but also the IV are a part of psychometrics. Okay, so when we are evaluating psychometrics of any study, we are evaluating the validity and reliability. Validity is of two types, internal and external. So internal validity and ecological validity, and reliability is of one type. So consider these things. When it comes to the IV, mainly take into account the validity. You can take ethical considerations also when it comes to the IV and DV. So that is one more area which is applicable. But first, most important, your focus should be on how valid and how reliable the IV was. In this case, if we take the IV, it was very reliable because it was a presentation of photographs of varying levels, which was standardized in previous studies. They were piloted, they were standardized because they were taken from an internationally established set of photographs with known valence and arousal. Such an IV, which is highly standardized and piloted, is obviously going to bring us reliable results. So that is a strength of this IV. Again, we have to make it specific to the study. This is how we make it specific. We mention the set, we mention the name of the set from which the images were drawn, and mention that what was the range of valence and arousal. That would make for a very good answer if that is how we write it. But in terms of limitations, whenever internal validity is high, external validity is low. Whenever reliability is high, external validity is low. So for any laboratory experiment, in fact, blindly you can write that the ecological validity is low. To that extent, you can write blindly, but then you have to make it specific to the study. So then you become alert and note why the ecological validity of this particular stimulus is low in this study, meaning the picture sets. What is so not ecologically valid about using these images? In the real life, when we experience emotional arousal or valence, that is when we experience emotional intensity, our experience of emotions does not come from looking at static images, no matter how standardized they are. In fact, in the real world, emotions are very unpredictable. The stimuli from which we feel emotional are of a variety of types. So we feel emotional looking at what other people do, just looking at some people, not even their activities, by looking at certain films, videos, by thinking about certain things. So there are a lot of stimuli which make us emotional in the real world, and hardly any of them are static. Most of them are dynamic. So definitely ecological validity is poor in the way the IV has been implemented. If instead of using photographs to make people feel emotionally aroused, the researchers would have used some real life stimuli, like I told you, relationships, thinking about something, or watching some films, that would have made the study more ecologically valid. So definitely in this study, ecological validity is low because of the way the IV has been operationalized. Again, my warning to students, do not assume that there is a limitation in the study makes the study useless, or now the study has no value because this is a limitation. Of course not. There are so many strengths of the study. It's just that the study is limited to this extent that we do not know whether its findings are applicable to the real world or not. Again, we do not know. We cannot say with certainty that the ecological validity is low. It is likely to be low, but we need to conduct a separate study, set of studies with more ecologically valid stimuli, and then we can check whether we get the same findings in those studies or not. So it is limited to that extent. It's not useless. Next is dependent variable. A dependent variable, how do we identify? We just ask a very simple question. What was measured in the study? And for all practical purposes, you can look at the results section of the study to understand which were the dependent variables and how they were operationalized. Directly, you get the answers from them. Coming to dependent variables, very important part of the study, because right from the aims, the researchers' focus is on the dependent variable. In this study, there were two dependent variables. Again, in the results section, clearly you'll see there are two different types of tables and graphs. So of course, right from the beginning, we have been seeing activation of the amygdala was the researcher's concern. How can we measure activation of the amygdala? We can measure for how long it is activated. We can measure how intensely it is activated. We can measure its frequency of activation. We can measure, like I told you, its duration of activation. So there are a number of options again to measure the activity of any part of the brain. What did the researchers choose to measure in this study? In this study, how did they measure activation of the amygdala? That will answer the question of operational definition. So in this study, the researchers chose to measure the activity of the amygdala in terms of percentage activation of the amygdala in response to participants' ratings of pictures. What is a percentage? It's just a way of expressing frequency. How many neurons were active, meaning I told you last time chemical exchange was taking place in how many neurons of the amygdala, and expressed in terms of percentage for easy understanding, for easy interpretation. So that is how the researchers operationally defined the dependent variable, first dependent variable of the study. In terms of strengths and limitations, there's hardly any limitation to this measurement. Why? Because it has been measured through a very sophisticated technology, fMRI, which we will not doubt in terms of the results it is giving us. It's too sophisticated to give us inaccurate measurements. So there's only a strength of this type of operationalization, that the measurement was highly sophisticated, simply meaning that validity is going to be high of such measurement. Accuracy, validity is going to be high of such measurement. So when you note it as in an answer, you write the strength was that the measurement was highly sophisticated because it was done using technology which resulted in the percentage, that technology which is known to be highly accurate, and it's called the fMRI scan. This is how you can write your answer very specifically. Now coming to the second DV of the study. The second DV was the memory for pictures shown in session 1. Right? We already saw in the procedure, participants had to look at new and old images and tell the researcher which were new and which were old images. So your memory for pictures in session 1 means, how was it operationally defined? Again, researchers could look at it differently. You could take a recognition test like these researchers did. Instead of that, there are other ways of measurement. Instead of just asking participants to rate in terms of new and old and give confidence ratings, instead of that, it could have been restricted to just being rated as new or old, or it could have been just sorting out the images in piles, or just giving a rough estimate of how many photos were seen, how many were not seen. So again, there are different ways of measuring this same variable. For the operational definition, we look at in this study how this variable, memory for pictures, was measured. So in this study, it was measured in terms of a percentage. So again, it's a way of expressing frequency of pictures recalled. Actually, this is a mistake here, so please correct this if you are noting from here. It's not percentage of pictures recalled, it's a percentage of pictures recognized. I hope you have learned in general psychology introduction when it's given to you. I hope you've been told what's the difference between recall and recognition. Recall is when you just remember something without any cue. Like when you write an essay answer in your paper, which you do. So, and even a short answer, for which there's no cue, you just remember from your own memory. So if in your paper they're asking you about what were the results of a study, they don't give you any cue, and they don't give you that multiple choice type of format. Just pick up which was the result of the study. You have to remember by yourself. That is known as recall. Recognition is when all the options are there in front of you, the correct answer is also in front of you, but it's mixed with other answers and you choose which is right and which is wrong. So this was, in this study, it was recognition. So it was a percent of pictures recognized because the pictures were there in front of the participants. So in terms of forgotten, familiar, and remembered from session 1. How it has come down to forgotten, familiar, and remembered? Because if an image was indicated as being new, whereas the participant had seen it before and in the second session he could not remember, it would be taken to be forgotten. Familiar and remembered ratings we have seen before, from where they are coming. So for photos which participants did remember seeing, they had to indicate whether they were familiar with them or whether they remembered them or they were just familiar with them. So these three possibilities were there of responses on this dependent variable. So this is the way it was operationalized for this study. Now, what are the strengths and limitations? Here again, I told you have to think in terms of psychometrics. When it comes to the dependent variable in terms of psychometric, yes, we can talk in terms of reliability, validity, but also we can look at the way measurement was done, in the sense of whether it was a quantitative measurement or qualitative. Quantitative measurement is again a concern of reliability. So it's within that only. And qualitative measurement tends to make the study unreliable. So here where I have noted self-report data. That is a type of qualitative measurement, though it may be expressed in terms of quantities, but it tends to be qualitative. So definitely when it comes to dependent variable, we will focus a lot on reliability. Within that, we know there are quantitative and qualitative types of measurements. So those issues we can discuss. Coming to the strengths of this dependent variable, it was quantitative measurement, definitely, because it was a recognition test, and we see that only 3 possible classifications were there. So any researcher could easily assign the number 0, 1, 2 for this type of measurement. So take forgotten to be 0, familiar to be 1, remembered to be 2. So the variable was easily quantified. It was measured in quantifiable terms. Why is quantifiable measurement good? We have linked it to the study, but we also need to say why it's good for a complete answer. See, when you're writing an answer, you need to mention which strength you're recognizing in general. You need to connect it to the study, like I told you, but you also need to say why it is a strength. Okay, so if you're saying quantitative data, yes, it's a strength. You're connecting it to the study, very good. But why is it a strength? Why is quantitative data a strength? Because quantitative data, when it is interpreted, when numbers are interpreted, they have some assigned meanings with them, which are decided beforehand, before the results are actually computed. Okay, so researchers first only they decide that if the participant gets, say, a score of 5, it indicates this. If he gets a score of 10, it indicates this. That means after they have measured whatever they are measuring, they cannot interpret it in biased ways. They're going to interpret it in prefixed ways, and they have numbers in front of them, not words, so they cannot mix up their interpretation with what they have found. Simply saying quantitative data is free from researcher bias to a very large extent, which makes results more reliable and valid. Okay, so that's why quantitative data is advantageous. Again, something you've learned before, so you should be able to express it in your answer over here. Next thing, in terms of limitation, it was self-report data. Some students feel self-report data, the only problem is social desirability. Again, that's a mistake. Self-report data has a lot of problem inherent in it. So here, maybe participants were not being socially desirable. But when it comes to self-report data, one very big problem is participants sometimes in their head, because self-report data means they are saying something from the way they are processing it. So when they are processing something, there can be confusion. They can be unsure about certain answers. But because it's self-report, they feel forced to select one of the categories which is presented to them. So if you're asking participants, have you seen this photograph before or not? You are giving them a forced choice. Either you say you've seen it before or not seen it before. You're not giving them an in-between option. Okay, I'm not talking about stage 2 where they could indicate familiar or remember, the very first stage where they had to say they've seen it before or not. Okay, at that time they may be confused in their head when they're answering, not very sure, and then they feel compelled to choose one of those 2 options, and there they can make an error. Okay, so that problem is also then self-report. It's not just about social desirability, it is about whether participants are able to express themselves correctly or not. So that becomes applicable to this study over here. Okay. If such confusion is there that participants actually remember something, but just because of the confusion they are feeling, they have indicated they don't remember it, that confounds results. Because despite remembering, they are not able to indicate that in the findings. So that's why we will say that this is a limitation. It reduces the validity of the findings because it introduces confounding. Okay, coming to the next element of the method section, which is controls. Controls you do not evaluate because the purpose of controls is clear. How do we identify controls in a study? First, we identify the IV and DV, like I told you. Then we need to consider which factors other than the DV, other than the IV, could influence the DV. All of those account for confounding variables. those confounding or extraneous variables, as they are called, are controlled by the researcher to make sure that nothing besides the IV influences the DV, which would result in valid results. So that is known as controls or control variables, right? So how do we identify controls? We just ask ourselves what all in this study could have influenced the DV, which was held constant or made sure by the researcher that these things do not influence the DV. We need to identify such things from the study. Again, for exam purposes, I think even if you remember 2 controls, that will suffice from what I see in past papers. Try to remember 2 to 4 controls. Don't need to remember all. But whenever you remember a control for a study, very important, you should be able to justify why the control was implemented. If you're just going to state a control in the paper, you're not going to get full marks. You need to justify why that control was necessary. So when you're preparing your list of the controls of a study, you not only state and name the controls, you also include with that a justification of every control, like I have done here. So let's take it as an example. Now, controls are implemented at all levels of the method, all stages of the method, right from sampling till the end, which is data analysis. Everywhere you will find controls. So see how many you can identify, how many you can justify correctly, and note those. So again, I have given a long list here. I told you the video is going to be long. So coming to controls, the sample first. In the sampling, the control was that all participants were women. This was a control because you can see when I've listed for every element, every control, I have listed the justification. This was a control because women are known, this justification is given in the paper itself. Women are known to be more physiologically aroused when emotionally stimulated. How does this help? Because obviously the fMRI scanner will be able to easily detect the amount of arousal in a woman, because her physiological symptoms are obvious when she's aroused. If males were used instead, again do counterfactual thinking like you do for standardization. If males were used instead, even if they're emotionally aroused, it's not very prominent. So the fMRI scanner would not be able to detect that activity so clearly as it would be able to do in the case of women. Okay, so very prominent and clear results would be found in the case of women. And this is why it becomes a control, because otherwise the gender of the participant would become a confounding variable, because for one gender, emotional arousal is not strong enough for clear distinction, but for the other gender, it is clear distinction. So it's better to use that gender to get valid results, to get more clear results. We are not saying this is not a limiting factor in terms of generalizability, it is, but in terms of controls, it's a plus. The same aspect of a study can be a plus in one way and can be a minus in one way. So in terms of control, this is a plus. In terms of generalizability, it is a minus. Okay, so this was one control of the study. Clear detection, so that the ability of the fMRI to detect arousal, okay, and to distinguish between different levels of arousal was not confounded by the gender of the participant. That's why this control was kept. Next, you can see randomization of picture across participants. Anytime there's a randomization in a study, that's a sure shot control that is being kept. Whether participants are being randomly assigned or whether a condition is being randomized across participants, which is the case in repeated measures type of studies. It is a plus, it is a control. Why is it done? Because order of exposure can influence the IV-DV relationship. What we mean for this study is, if pictures were not randomized and say the pictures were shown in a certain order. So first participants were shown all very less arousing images, very less intense images, and then moderately intense images, and then very intense images. This order could have influenced the amount of activation of the amygdala, okay, because too many arousing images together, too many less arousing images together, that could have hampered the way the amygdala was being activated. That could have hampered the way participants remembered images, because you know, when something is shown in order, participants tend to remember the first half and the last half better, the serial position effect. So all these things could have got influenced if the images were shown in some particular order. So just to prevent order of exposure from confounding how aroused participants felt, how much the amygdala was activated, and what type of memories they formed, that's why the pictures were randomized for every participant. Next is that fixation cross, which was there between pictures in that interstimulus interval for about 13 seconds. Why was there a fixation cross? To prevent carryover effects from one trial to the next. One trial in an experiment, in this experiment meant that one exposure of one photograph, that was one trial. So if participants see, say, a very emotionally arousing picture, and because of randomization, the next picture is not so emotionally arousing, what would happen was that lasting effect of arousal would have extended to the next trial also, to the next picture also. If you're feeling very emotional and seeing one image is an image and then I replace it with another one, your emotion doesn't die suddenly. You still feel very aroused even when you're seeing the next image, but not because of that image, because of the earlier image which you had seen. So to prevent that carryover effect, there was a fixation cross, a blank screen and a fixation cross, to make sure participants focus on the screen instead of getting distracted or keep on ruminating about that previous photograph, so that their mind would be cleared of the emotion they had felt for the previous photograph, and then the next photograph was introduced. So that was the way that fixation cross was introduced in the study. Also keep in mind, when the researchers decided that the fixation cross will be showed for about 13 seconds, definitely they must have had some logic. There must have been some findings in past research which suggested that 13 seconds are enough to clear off the emotional baggage of the previous photograph which is shown. So they must have taken an informed decision to maintain that fixation cross for 13 seconds. Next is use of research established stimuli. Like we saw before, standardized images were used. Again, because if the researchers think counterfactually, if they had prepared their own photographs or just selected any photographs which was easy to obtain, in that case that quality of the photographs would have influenced how the amygdala was activated or how participants remembered the photographs. Okay, so if some photograph was very vague, very weird, participants would have remembered it better even if it was not emotionally arousing. So to avoid these type of things from happening, they chose standardized photographs known to have certain emotional properties. Next, we have foils were matched in emotional arousal. We saw that earlier also. They were matched with the true pictures using significance tests. Okay, so significance tests were used. Significance tests just tell us whether whatever is there in our sample is true for the population also or not. Again, I won't go in the details of this, but these significance tests showed that the foil photographs were significantly matched with the original photographs. This control was very important, because if the foil photographs were different, imagine they were markedly different from the original photographs in terms of emotional properties, in that case participants would have easily identified which they had seen before and which they hadn't, because these would be very distinct from the earlier ones. So we wanted them to be able to genuinely express which they had remembered and forgotten, and not just because the photos were looking drastically different. So this was one more control. Next, you can see in the scanner also some controls were maintained. I told you earlier also, unnecessary movement was going to confound results. If there was some unnecessary movement of participants in the fMRI scanner, the scanner requires that participants should be still. They should be calm and still when they're inside the scanner. Calm in the sense not emotionally but physically. Okay, so outwardly there should be no movement, so that the fMRI can accurately detect their state. That is why what was done to minimize the head movement, because you know what happens when there's a strong magnetic field around 1.5 Tesla magnetic field, the head tends to move. Okay, head can't remain still. So what is done is a bite bar was given to participants. Bite bar, you know. Okay, so that was put on the head of the participants, in the mouth of the participants, so that their head would not keep moving. That to bite on that bar hard so that their head would remain still. Okay, so this prevented unnecessary movement in the scanner. So again, it's a control, otherwise movement would have determined activity of the amygdala rather than the emotional value of the images. Next is that strong correlations were established between arousal and balance of photographs to determine and participants' ratings of emotional intensity. When I told you that the way the researchers selected the images on basis of valence and arousal, that is what determined how participants perceive the images. How are we sure of that? How do we know that pictures which were known to have high arousal and low valence were the ones which participants found more emotionally intense, and those which were known to have low arousal and high valence were the ones that participants found to be less emotionally arousing? How do we know that? Because correlations were established. So statistical analysis was done after the research was conducted between the 10 participants' ratings for all the photographs and the known arousal and valence properties of the photographs. Correlations were established. I hope you know correlations. So they were established and they indicated, the figures also there, I think you need to remember, I've missed out on writing on them, something like minus 0.68 and plus 0.66. Such correlations were found. These are strong correlations. They are close to 1.66. It's 0.66, 0.68 is close to 1. So such correlations indicated that definitely participants' perceptions of the photographs were matching with their actual emotional properties. Okay, so researchers did not just assume this, they knew this from statistical analysis, they had proof of this. Okay, so that again is a control. Otherwise, if their manipulation had failed in the sense that they had shown certain photographs of the participants had interpreted it or perceived it totally differently, entire results would have gone wrong because of that. It would be a major confounding. Okay, if researchers had chosen highly emotional images but participants perceived it as not arousing at all, then the entire experiment would have gone haywire because of that. Next, we have the final control which I have noted is that approximately equal number of photographs were determined as belonging to each category of emotional intensity. So we had 96 photographs, right? And each of them were rated by participants. So if you divide 96 by 4, roughly whatever number you get, that many images belong to each category. So that many images, like in terms of percentage, it has been presented in the paper. So roughly 25% of these 96 pictures belong to those 4 categories, 0, 1, 2, 3. See what would happen, again counterfactually, if different numbers of images belong to each category. So imagine out of 96 photos, 50% of the photographs were found to be emotionally intense by participants and 0% photographs were found to be not intense at all. Then in that case, whatever results we would get, they would be because of certain photographs were present. So obviously ratings for them would be higher because there were such photographs, and certain photographs are not there at all, so ratings would for them would be 0. So the number of photographs would determine participants' ratings rather than their emotional properties. So again, this was not the case. Researchers established it with the help of statistics. They actually counted and came down to the percentages of how many photographs belong to which category. And they found that these were roughly equal. Okay, so whichever controls are very clear to you in any study and which you can justify fully, prepare those for your exams. Next, we have experimental design. Experimental design is directly determined by the independent variable 1, and secondly, the participants who are used for the levels of the independent variable, how they are used. Okay, independent variable is the first thing. If you have not identified it correctly with its levels, you will not be able to identify the experimental design. And how you justify the experimental design is always in the paper. You state which was the independent variable and how many levels it had, then only you will get full marks. So in this study, definitely it's a repeated measures design. You know, students tell me very easily what's a repeated measures design. Same participants were exposed to all the conditions, yes. But when you're writing in the paper, you state which were the conditions. So you state the IV, its levels are the conditions, you know, and then you say that same participant was exposed to all these 4 levels. Okay, so that is how you justify. Also, you should, for good practice, you should say because these were the 4 levels of the IV, so these 4 groups were created, or these 4 conditions were created. This is a repeated measures design, so conditions will be created, not groups. So here you can see, so 4 conditions were created ranging from no emotional intensity to very high emotional intensity. So participants were exposed to 4 conditions. In one condition, there was no emotional intensity, then mild, then moderate, and then a lot of emotional intensity. So these are the 4 conditions every participant had to undergo, and that's why it's a repeated measures design. Coming to the strengths of the design, when it comes to experimental designs, in terms of which strengths and limitations you should be looking out for, what you've learned in your research methods. Okay, for every design, be it independent groups, repeated measures, or matched designs, you have learned strengths and weaknesses. So those you will have to remember and directly apply here. Mainly these have to do with what are known as order effects and practical considerations. Okay, but then I would say there's nothing so general that I can identify here. It's best if you learn those properties from the research method section. When it comes to repeated measures design, the main strength of a repeated measures design is what we call high statistical power, but that terminology is a little advanced for you all. If you understand high statistical power, good. If you don't, you can just remember that participant variables are eliminated from repeated measures design. When we have an independent groups design, participants' own variables, or they are what we call individual differences. Some students are more familiar with that term. So individual differences can account for the results that we find more than the manipulation done by the research. That is not a reality when it comes to repeated measures design. When it comes to repeated measures design, participant variables are eliminated because the same participant undergoes all the conditions of the IV. So in this case, for Canli et al. Study, we need to ask ourselves which were the participant variables which would have confounded the results, then we can make it specific to the study. So for Canli et al., the participant variables that would have influenced the results of the study were two, because there were two DVs. One was the emotionality of participants, or you can also say amygdala, to what extent it's activated. It means the same thing, but you can present it either way. So everyone has a unique level of activity in his brain structure is also unique. So that would have been one participant variable that would have influenced results if independent groups design was used. Another, the memory of participants, memory capacity or memory strength. So both of these were held constant in this experiment because it was a repeated measures design. So you can note that when you state this strength. Next is there's a need for a smaller sample size. This is a practical consideration. Smaller samples are easier for researchers to recruit. For this study, why a smaller sample was more desirable, we've already noted in the sample section. It's a biological study with very expensive technology, very effortful study. So definitely there was a need for a smaller sample size, and more results can be found with smaller samples when it comes to repeated measures. Otherwise, there were 4 conditions, so at least we would need to make 4 groups. So we would need 10 into 4, 40 participants, which would be difficult for the researchers to manage. So in this case, having 10 participants was suitable to the experimental design, and it made things easier practically for the researchers. Coming to limitations, order effects are a problem in repeated measures design. Some students tell me order effects are same as fatigue and practice effects. No, those are types of order effects. An order effect in general means the influence of a previous condition on the coming one. That is an order effect. It can be in terms of practice, so that's one type. So because participants have undergone a certain order previously, they have practiced a certain task, and now that practice is influencing the upcoming condition. So that is one possibility. Another is fatigue. They have practiced, they have undergone certain condition before. They're tired because of undergoing that condition. So that is influencing their performance on the next condition. So these are just types, but actually order effects is the broad category within which we have practice and fatigue effects. So when you're saying, you learn in the preliminary section, students learn that repeated measures design suffer from order effects, but you need to see which of these order effects, practice or fatigue, is applicable to this study. You can even include demand characteristics in this. That is also a type of order effect, in the sense that when a participant comes to know about one condition of the study, he might guess the next condition. Or when he undergoes 2 different conditions of the study, he understands the purpose of the study, because he sees the contrast between the 2 conditions. So in one condition, when you show him very emotional images, in the other you show him neutral, he understands that this study has something to do with emotions, because here I'm seeing emotional images, then they are not showing me emotional images, something it has to do with the purpose of the study. So that type of understanding comes in the participant. So you can include demand characteristics also in order effects. So for any particular study, see which type of order effects apply. Do all of them apply? So practice, fatigue, demand characteristics, or one of them applies? You need to look at that. So coming to order effects, for Canli et al. Study, which would have applied? What you need to imagine is, if instead of randomizing photographs, participants were shown all intense photographs first, then mild photographs, then even milder, then what would have happened? Okay. Or even if it has been randomized, what problems would be there? I'm sorry, here what I've done is, in the limitation I've included order effects. It should have been in the strength. So please correct that. In this study, it was randomized. What we need to imagine is, if it was not randomized, then which order effects would have played a role. In the Canli et al. Study, practice effects would not take place, because emotions are not a task that participants perform. So if we were to give them images in an order, they would have not been able to practice how much emotion they experience. Nobody can practice that. So practice effects would not apply. fatigue effects could apply because watching too many arousing photos or intense photographs together would have made participants tired. So fatigue effects would apply. Demand characteristics could also apply in that case, because participants would see that initially they are shown very sharp images in terms of emotions and then little duller images. So they might have guessed. But again, that would have not mattered because they could not control how active the amygdala was. So more or less fatigue effects would have counted in this experiment. Okay, which could influence the way the amygdala works. When you're tired, your brain works in a lower capacity than when you are not. So that would have been a possibility had the order not been randomized, but it has been randomized in this study, so this counts as a strength, not a limitation. So I'm sorry about that. Next is repeated measures design are more time consuming. Okay. Yeah, one more thing before I come to this more time consuming aspect. If some possible limitation is there, like of an experimental design, which we saw your order effects, if that has been controlled successfully by research, he's done something to account for it, it can be included as a strength of the study. Okay, next, coming to the next limitation, which becomes the only limitation here of the repeated measures design, it's more time consuming. Yes, because what happens in the case of independent groups design, you can run many conditions simultaneously. So if we had separate groups for intense, less intense, lesser intense photographs, we could have run the experiment simultaneously with all these groups and it would have got over quickly. So again, a practical consideration, but when it comes to repeated measures design, because the same participants have to undergo different conditions, they can't do it together, so it becomes more time consuming. Overall, if you see the Canli et al. Study is not very time consuming, frankly speaking, because few seconds for every photograph and only 96 photographs. In the later section also 96 plus 48 and few seconds for every, just till they recognize the photographs. Not very time consuming, but more time consuming than an independent groups design would have been. So in comparative terms, we are saying that it is more time consuming. So this is about the experimental design. Then we have only two aspects left. One is ethics. Ethics is the best section. Even students find it very easy when it comes to method, because you just need to you just need to check every ethical guidelines. You can prepare a checklist of ethics and you just have to check whether it was followed or breached in the particular study. So here also I have presented a list. It's best to follow this list and just check if every ethic has been followed. When it comes to ethics, in many studies you will have the problem that you are not sure whether an ethic has been followed or breached because it's not spoken about at all or not even implicated at all. In that case, you can just mention that it is unspecified, and the examiners are unlikely to ask such questions. They know it is not there in the paper. They don't want to waste time discussing those. But if at all they ask you about those, you can say it's unspecified in the report. Okay. Sometimes you have to infer ethics. Okay, just because it's unspecified does not mean you write unspecified because directly they have not spoken about the ethics. Sometimes you have to infer. Okay, so make sure you're not even able to infer it and then you say, okay, it's not specified. Coming to the ethics, the first one always is informed consent, because that happens right at the time of recruitment. Here we can assume informed consent. Why? Because the sample was voluntary. Volunteers, when they come, they know why they're coming. Yes, what we need to check in informed consent is to what extent they know about the purpose of the study when they have come in. So how much do they know? What have they been told is the purpose of the study? So when the study was advertised for, or when they came to the laboratory, what were they told? Why is the study happening? That we need to consider, but this is something we do not find a mention of in the Canli et al. Report. So we can assume that there was some informed consent, but the extent to which informed consent was there, we don't know. Next is the right to withdraw when it comes to ethics. This also can be assumed because volunteers, when they have come in, they know they can leave the study at any time. They might assume that they have to stay till the end. They can't leave it midway. Also, if they are in an fMRI scanner, they cannot just get out of it suddenly. And they had a bite bar and they could not really say that they wanted to quit the study. So to that extent, yes, right to withdraw was not there. But why I would say it is assumed is because when volunteers come in and they sign a consent form, there it is specified that, you can leave the study at any time. So if they're asking you in the paper, you can give a balanced view on this. You can say for these reasons, you feel that it was there, but for these reasons, it seems it was not obeyed. So you can give both sides of the picture. Next, it comes to deception. There was no deception in this study. In the sense, the real purpose might have been concealed to some extent, but the participants were not misled in any ways about what the study, because you cannot mislead them. They are in the fMRI scanner, the images are in front of them, so you can't lie about it to them. What is happening in the study. Their brain is being scanned, they know that. They are inside the scanner. They are seeing emotional type of photographs, they know that. They are right in front of them. Their memory is being tested, they know that, because they are being asked about what was shown in session 1. Yes, there was, I think it is mentioned that there was an unexpected memory test. So to that extent, concealment was there of the purpose and what was to be done ahead, because they did not want participants to intentionally memorize images. That was there, but there was no misleading of any type. Again, you can give a balance view. So you have written there was no deception, but if you want, you can consider both sides. Look at what question is being asked to you in the paper and answer accordingly. Next is privacy. Privacy was maintained. We noted right at the beginning it was individual testing, and participants' brains were looked at, but they were not looked at in the sense nobody was staring at them from a one-way mirror or from a camera or something. That was not happening. So their brains were being looked into. That's not an issue of privacy. Also, they were individually tested. They were inside a very confined scanner. So definitely in all these terms, privacy was maintained. Confidentiality was also maintained. How do we know confidentiality is maintained or not? We look at the report. How many details have been revealed about participants in the report? So in the report for Canli et.al, we see the gender of participants overall is given, not for any specific participant. So all are female. So yes, we can say any one particular participant, we know it's a female. That is told, and they were volunteers. They don't even tell us from where they came. So definitely a lot of confidentiality is maintained. There are no details of any participant that could help to identify that participant by name or by identity. Okay, then coming to harm. Harm was there in this study. There was a possible physical harm in terms of the fMRI scanner being there. Again, students tend to exaggerate this a lot when they consider ethics in the paper. Please be very realistic when you are considering the extent of harm involved in any study. Okay, just because if it was an fMRI scan and there was a magnetic field and they were in a tunnel, you don't behave like as if they were going to die inside that scanner. That was not going to happen. Researchers knew it was very safe. So we say there was a possible minimum harm. Yes, something could have gone wrong, or some participant, despite them having seen to it that participants were healthy. See, if you're writing about harm, mention that they had checked, they had verified participants were healthy. So they did not take that risk of putting unhealthy participants and, hampering their health. They did not put unhealthy participants into the scan. Please note that. Take a balanced view always. But at the same time, always there is scope for some harm coming up. For, researchers did not know that someone had a certain problem. The person himself did not know he had a certain condition and he went under the scanner, or the scanner failed in some way. So some harm is there, definitely, but to a very, very, very small extent. So minimal harm. In coming to emotional distress, that was high in this study because some of the photos were highly emotionally intense, like we saw. So they were very arousing also for participants, mentally and physically, and they were very negative also, some of them. So valence was very low for some of the images. So in that case, yes, participants must have gone intense emotional discomfort. Whenever we talk about psychological harm in the paper, we need to note whether anything was done to rectify that harm. Okay, for like in the case of physical harm, we saw that it was assured that participants were healthy. So that was a preventive measure taken to make sure that they were not harmed. What about psychological harm? What was done for that? So we need to check if any counseling was done for participants, if during the process of debriefing, anything was done for them, they were given an opportunity to vent out, or some procedure like, showing them positive images to make them happy, to arouse happy emotions in them. If that was done, we need to check for that. So what was done to account for the psychological harm which was known to be caused to participants? In the report, nothing is mentioned. So it might have been done, we don't know, but because it's not there in the report, we say that nothing was done to account for the harm. So there was scope for some counseling to be offered or some attempt to be made to reduce the amount of discomfort that participants felt. And then we have debriefing, another ethical concern. We don't know. It's not mentioned in the report. It's likely to have been done. Most cases it is done, and this is a recent study. Recently, ethics are followed a lot. This is a study from 2000, not very recent, but recent. when ethical practices have come in quite soundly. So it is likely to have done, but because it's not mentioned, we don't assume that it was done. We just say it was not done. Okay, and then use of animals and children. Okay, one more thing, sorry. When it comes to debriefing, you need to note whether it was necessary or not. It was necessary in this case because psychological harm was caused. So if they ask you about debriefing, you can connect to the psychological harm. So some debriefing, some opportunity to participants to question, to vent out, and some explanation given during debriefing would have been very valuable for them because they had undergone emotional distress. Then coming to use of animals and children, no, they were not used in this study, so this ethic is not applicable here. As for use of animals and children, students get confused in the issue of use of animals and children and the ethic of use of animals and children. So when we come to issues and debates, which I'll make a separate video on, in that I will talk to you about how you consider use of animals and children potentially in any study. Here, when we are talking about use of animals and children, we are saying whether they were used or not. We are not talking about what if they were used. That we are considering in issues. Okay, and then coming to data analysis. Data analysis is not included in the syllabus as an element of the study. Because in results you come to know, it becomes very obvious how data were analyzed, but still I prefer to cover data analysis before transitioning from method to results. Okay, so coming to data analysis, simply saying how the data obtained, how information or quantitative, qualitative data obtained from participants was analyzed or processed for obtaining results. So in terms of data analysis, when it came to activation of the amygdala, see we can go DV wise. So one DV was activation of amygdala. How was the data from this measurement analyzed? So when participants underwent the functional scanning in which the amygdala activation was noticed, 11 frames were captured per trial. One trial means exposure of one image, like I told you. So every time participants saw one image, any one image, 11 frames were captured, means 11 photographs of their brain was taken in its active state, because this is a functional scan. 11 frames were taken. Why 11 frames were taken or 11 photographs of their brain for every single image that they saw? Because of the hemoglobin response. Please see the introduction video if you don't understand this. That hemoglobin response, which is very important, that takes place after 4 seconds. Okay, so that's why researchers wanted a baseline for every image. So what is happening, the first and the last 2 seconds they took as a baseline. Baseline meaning when emotional activity is not taking place at that time, what is the state of the amygdala for this particular participant? How active is the amygdala when there is no emotion? That they wanted to capture. So that they came to know from the first 2 and the last 2 frames out of the 11 frames they captured for every image, and from the middle frames 5 to 8, from that they came to know about the emotional response, and then they could contrast it with the baseline response because they had collected both. So after the image is shown when the amygdala starts resting, and before the image is shown when the amygdala is at a rested state, those baseline frames were also captured, and during the activity also the frames were captured. Okay, so this was the way activation of amygdala was measured. This was how data was analyzed for amygdala activation. And further, like I told you, they subtracted the baseline activity, baseline percentage activity of the amygdala from actual emotional activity of the amygdala, and they came down to results for every single image for every single participant. So that is how intensely data was analyzed. And what they did then, we know, we have seen earlier also, they calculated correlation coefficients between whatever ratings the participants gave, whichever button they pressed, whichever number they indicated, and the emotional and the arousal value of the images which they knew. So they correlated that. Okay. Also, they correlated the activity of the amygdala with the button which was pressed by the participant. Obviously, now why they'll do that? Because they were expecting that there would be more activity when a higher button was pressed. So when 3 or 2 was pressed, there should have been higher activity in the amygdala. So to check whether this was really the case or not, they calculated the correlations. So this is how they analyzed data for the first DV, which was activation of amygdala. There was another DV also, we know, memory for emotional images. How did they analyze data for that? They had, first they computed the mean ratings. Again, you know means and averages from your preliminary section. So they calculated mean ratings for all those 3 responses given by participants: forgotten, familiar, or remember. So they calculated the means for each. And then they conducted a repeated measures one-way ANOVA. Repeated measures because experimental design was repeated measures. ANOVA because the IB had more than 2 conditions. There were 3 conditions. So that's why ANOVA was calculated. And if you don't understand ANOVA, I'm sure you understand that it is a type of significance test. So a significance test suitable to this study was done. That much you can understand. And that was done for memory performance, and it was found, we will see later, that there was a significant difference between these the memories for these 3 different emotional intensity ratings. As of now, this is what we see for the data analysis. Okay, so today we have covered a lot of ground in terms of the method. The method is the lengthiest section, most important, and as a psychologist, where you need to have the most skills is the method section. Not that you don't need to have skills to form an introduction or calculate results, but yes, or to discuss results rather, because calculation you do in the method. But yes, method is the section where you need the most skills. So as a student also, you spend the most time in the method. Prepare your method very well for all the studies. This is where students lose the most marks. They are not able to operationally define variables, not able to give the strengths and weaknesses of different elements. And it's sad because easily they could have prepared for it before the paper. So spend your most time here and keep revising what we've done today. Try to extend what you've learned today to other studies, and I will come soon with the results and conclusion, which will be the next video for this study. After that, there will be a video on the research method laboratory experiment and the approach, which is the biological approach, and finally there will be a video on the issues and debates. So all of that I will come with very soon. Till then, keep practicing how you should be going about in the method section of any study. I'll see you soon. Goodbye.